So it's a bit different in the fetus compared to the pig after it's born. So it's not the lung that is the primary site of virus replication. So once the virus reaches a fetus, it mainly replicates within lymphatic tissues.
Hello, welcome to this edition of Meet the Expert, a new series of podcasts on swine disease management in practice presented by Boehringer Ingelheim. My name is Peter Best. You refer to the genetics of the host in this study. Is this in relation to the potential for genetic resistance to be identified and bred into the animal, or was it not part of that?
Yeah, that was the whole idea behind it, to look at maybe not resistance, but more resilience. So to see that you breed for less susceptible animals that deal with the disease in a better way.
Yes. Now, I don't know whether it's from that study. I remember reading that some animals with a resilience, as you describe, can be identified even within their genetic code, but that was only true for certain types of virus, perhaps type 2 and not type 1 virus, or some variation in the virus.
I guess we have there are different groups doing different type of research in the host genome or in the genetic resistance of the pig. You might have heard about the PRRS resistant pig, and that's a different type of research in the end. So what we have done is infect a high number of pigs and look into so-called nucleotide polymorphism, so in variation in the genome that can be then associated with more or less susceptibility to the disease. And that you could use then in the future to select for less susceptible animals. But then on the other hand, you have researchers that look into the receptor of PRRS, the CD163, and try to modify that receptor so that the macrophage, so the target cell for PRRS, is not going to be infected with the virus, at least with those strains that we have right now. Because that's the whole story about target cells and susceptibility of cells to PRRS is quite interesting, I would say. And we don't know what happens in the future, because even if we have animals that don't have the CD163 receptor that allows the virus to enter, maybe in the future we have different cell types that the virus might enter when it keeps continuously evolving and changing. I don't know.
True, and I believe that the CD163, that original, was a very, very small study that they actually had a complete gene knockout of. So that's a haptoglobin. It's a transmembrane protein, I believe, that's a haptoglobin scavenging protein, and that it actually has more normal functions than even haptoglobin scavenging. And they found with the complete knockout that it also damaged the pig's normal functioning significantly. So they've been attempting to isolate just the part of that particular protein that's involved in the receptor function, and then only alter that so that the protein retains its other functions. And at this point, I think the story continues. Yeah. And I would have to say the same thing. We have a virus that has an incredible ability to mutate. And with all of these mutations of viruses, especially the RNA ones that mutate so much, mutations going on all the time, a lot of the mutations are fatal so that the virus cannot form again. But mutations by nature, it's the ones that provide an advantage are the ones that then become dominant. So take this example of removing the receptor with a virus that does that, and we certainly won't have all the pigs in the world immediately be CD163 negative pigs. Yes. So the virus will still exist in the face of pigs that don't have the receptor. I would have to think it'll find a way in.
Yes. I've asked you about the research level, but this podcast is aimed at the field veterinarian, the veterinary practitioner, and let's go back to the basics. What should a practitioner think about or be concerned about regarding high virulent PRRS forms? What are the major concerns from a practice point of view? And could I put that to you, Dr. Stevenson, first, please? If in your everyday work as a veterinary practitioner, what should a veterinary practitioner be watching, aware of, or looking for?
Well, yeah, and I'm an everyday veterinary diagnostician. I'm not an everyday veterinary practitioner. I wouldn't pretend to know as much as many of these swine-specific practitioners do in that way. But I think for them, being aware of the different ways in which these high-path strains manifest, especially some of the odd ways with nervous disease, for example, or understanding that we're seeing some of these strains cross the placenta in the second trimester, and what they may be looking at and just assuming is a breakthrough with either parvovirus or PCV2 as causing second trimester reproductive failure, they need to now consider PRRS as a possibility there. They may start seeing much larger amounts of, for example, strep suis septicemia, and it may be that they've got a new strain of PRRS that's behind that, causing strep suis, the same strain they've always had of strep suis, to be able to be more aggressive because it's the underlying PRRS virus that's reducing the number of macrophages so profoundly in the pigs that then they're able to invade. The macrophage being really the front line innate defensive cell against bacterial invasion. And so I think it's that just as they become aware and hear podcasts like this or find other information, that they kind of put these things in the shelf of their mind so that they're considering them if they see these unique things they may not have associated with PRRS virus before. I think most of them will know if they're seeing the same things they've seen, they're just worse. They'll just say to themselves, this is PRRS virus and it's more severe than I've seen before. And they may just do more of the same things or reach out to producers of vaccine and others to find solutions. But it's the odd manifestations, I think, that they need to be aware of.
So aware of the changes from the past. Don't assume that what was in the past is going to be typical of the virus of appearance today. And you've suggested that the evolution will produce more high virulent forms in the future, so that the possibility would actually increase with time. One would speculate anyway, that there will be more of these forms opening the doors or having an effect earlier in gestation or whatever it is. So from that point of view, I understand. Do you think, therefore, that they should be looking in their client herds to see whether there are any changes in manifestation of the PRRS picture, which are not as defined as you've suggested, but maybe subtle changes? Are there changes which are not yet of an order which is completely out of the normal, but they're trending that way? Are there changes in the clinical picture like that?
Well, that's really hard to answer. I guess the reason I say that is one of the hardest things we do where I am, and I'm sure where you are, is that you have people seeing things that they think are different than they've been in the past. One of the hardest things to do is figure out whether you have an agent that's been there before that is manifesting itself differently, and actually assigning causation rather than association is a challenge. It's a challenge. And the easiest thing in the world to do is be wrong by making assumptions based on association, and that there's no proof for causation. We have that happen every day, and it really does no one any good. We need to find proof and be sure of what we're talking about. And that's hard.
You are listening to Meet the Expert, a new series of podcasts on swine disease management in practice presented by Boehringer Ingelheim. If you would like to know more about the subject we're discussing in this podcast, additional information is available offline. Dr. Stevenson mentioned vaccines, and I want to refer to some work I've seen of yours where you did some challenge studies with a new vaccine, or new in Europe vaccine, which involves some high virulent forms, am I right? Could you tell us a little bit of what was the background to that study and what you actually did?
Yeah, I can probably tell you a little bit about the background of that highly virulent strain. It was a European type strain that all of a sudden popped up. That was in spring 2015, I guess. All of a sudden we heard about PRRS outbreaks occurring more and more often. And from the field, it was the feedback that they are quite severe. And then we tried to get our hands on that strain and also got it in culture. And then saw the same thing also in the lab, that it did grow to high titers. It really killed the macrophages in the lab quite fast. And then we did the first experiment, putting it back into pigs, because when you simply describe what happens in the herds, we also got the feedback from reviewers, yeah, but as long as you don't put it back in pigs, you cannot talk about a highly virulent strain, because in the field you have a lot of cofactors influencing the clinical picture, and simply from what happens in a farm, it's not enough to say this is a highly virulent strain. So we went back and put it into pigs. And did that together with a vaccine experiment. So we had vaccinated pigs that were challenged with that strain and non-vaccinated pigs. And we did that in a reproductive model, but we also started to do it in piglets in a respiratory model. And in the end, it really confirmed that, yeah, this is a more virulent strain, or a highly virulent strain, also from a classical subtype 1 Western European type, that causes more disease, more damage, causes reproductive disorders at the same level, more or less, than type 2 strains. And it can also cause respiratory disease in piglets. And with the vaccine studies that we have done, we found that the vaccinated pigs had quite good protection to generally conclude, I would say, yeah.
What was different about the vaccine that you tried? Was it a standard vaccine as far as you're concerned, or I understood it was a specific sow vaccine
we have done reproductive models with the sow vaccine, but we have also done challenge experiments in piglets using the piglet vaccine.
Right.
So we've done both.
Right. And with high virulent forms, you're getting plenty of evidence that the vaccines are giving protection even against the high virulence forms.
And this is the benefit of having a high virulent strain of the European PRRS, that at least you have your non-vaccinated animals and you see the disease and you see the damage that the virus is causing. And that's what you need in the end to also prove that a vaccine is doing its job, because when you hardly get any clinical signs or any damage from in non-vaccinated animals, then it's also hard to get the difference between vaccinated and non-vaccinated pigs.
Given our earlier discussion, at what stage were you vaccinating your sows?
We did it as the company producing the vaccine is recommending it. So vaccinate the gilts before you inseminate them, and then do a mass vaccination every 3 months. And that's in the end what we tried to do also in those gilts. So they were vaccinated twice before insemination, and then one time in around mid-gestation, which would naturally occur in a 3 to 4 month batch interval of vaccinating the whole herd.
Yes, yeah. And was that compared with the effect of piglet vaccination per se, whether sow vaccination, gilt vaccination, or piglet vaccination gave you different results?
In the sows, it's easier, I think, in the end, to get a good level of protection. With the piglets, it's more complicated, I would say, to get the good protection on a piglet level, and not have any problems any longer with PRRS in the nursery grow finishing area. It's probably a bit more complex.
For what reason? Sorry, would you just explain that a little bit more? Why is it more problematic in piglets? Is it a practical application thing, or
I don't know, maybe it's partially it's numbers of animals that we're dealing with, or the maturity of the immune system. It also has been shown that there is something like a not resistance, but the older a pig gets, the better the immune system deals with PRRS. So the younger the animal is at the stage of infection, the more damage the virus is causing. So maybe with more adult animals, it's easier to get a better level of protection than in very young piglets. Maybe that has to do with it.
But certainly vaccination, we can be assured from your results, is protecting even with high virulent forms. So I guess your experience in America in the field would show good results with vaccinated animals in the face of high virulence challenge, would they?
I'm probably not the best person to ask that question. And the only reason I say that, being in the position I am in the diagnostic laboratory, I'm not at the level where those products are being used every day. And increasingly, the majority of our swine are produced by closely held companies. And those companies increasingly don't share that information.
Yes. Okay. Well, that's fair enough.
In the end, a lot of the literature that you can read out there proves that in experimental settings, the vaccines are working quite well. But then when it comes to the field situation, it's a lot more complex and a lot more complicated. And I guess you also deal with a lot bigger number of pigs. So to all get them immune at a certain time, it's not that easy compared to an experiment where we have low individuals. We all know their immune status. We know which other infections are going on or which they are free of and so on. So in the experimental settings, it's, I think, a completely different story compared to out in the field.
Well, and I think also when you're doing studies in experimental setting, you're tending to challenge the vaccine with a single strain, your challenge strain. In the field, we're dealing with a wide variety of strains that we know, in the type 2 strains, they vary, the genome varies up to 30%. Just at a genomic level. Yeah. And we know of herds that have been vaccinated and we have the sequence of the strain that they've had in the herd, their field strain, and they've held pretty well and they've been holding, and then suddenly they'll have an outbreak as if they weren't vaccinated at all. And it'll be, you can see that there's a lot of divergence based on sequence. That doesn't give us any idea anagenically what's significant or not significant in relative to protection, but we can look at what happened. And I typically tell people, the dilemma that we face is vaccine companies can have a very limited number of strains that can be in a vaccine. And we have such divergence in the field in what strains are out there. At best, we can hope these vaccines protect against a majority of the strains. But there's going to be a minority of them that escape, a vaccine's ability to protect.
So the outliers will be rare, but they will occur.
Yeah, well, rare unless they're the one you have and you produce a lot of pigs.
Yes.
Then it's not rare to you. So yes, that's the issue. And that's why we have so many kind of other things that happen, with people trying to protect animals that may be not the best practice.
Dr. Ladinig, from your experience with the challenge of the specific sow vaccine with these highly virulent forms, what would you say from that would be the ideal for sampling in terms of diagnostics? Should we be looking at certain sorts of fetal tissue more than others? What's your recommendation at this juncture, please?
So it's a bit different in the fetus compared to the pig after it's born. So it's not the lung that is the primary site of virus replication. So once the virus reaches a fetus, it mainly replicates within lymphatic tissues. And it's not that easy to sample a lymph node in a very small, maybe aborted autolytic fetus. So the thymus is one of the easiest and also biggest actually lymphatic tissues that you can sample from a fetus or from a newborn piglet. So in our studies, we always went to look at the virus in the thymus, and you can also go for the diagnostics to pool them from more than one piglet. So we usually do pools of 5. Nice. And then you get a quite good result that is representative for the complete litter.
So take lymphatic tissue.
Take lymphatic tissue. Don't go to the lung.
No.
So the lung is not the best place in a fetus to look for PRRS. Probably it has something to do that it's not ventilated yet as it is after birth, and maybe there are different types of cells there. Yeah. So go into lymphatic tissue and collect the thymus.
And a pooled sample from 5 pigs would be a good thing to try.
In the end, it's, I think, important that you take enough piglets from a litter, because that's also something that our experiments showed, that there is high variation in the number of infected fetuses per litter. So you might have litters where almost all of the piglets are infected, and it's not that complicated to actually find the virus. But on the other hand, you have individual litters where you only get 1, 2, maybe 3 fetuses that get infected. And if you have a total of 14, 15 or whatsoever. So it's really important that your number is high enough. So you're better off taking a sample out of each of the piglets and do pools so that you get 2 or 3 pools out of the complete litter, but you cover all of them. Then your chances of actually finding the virus are way better compared to only sampling 1 or 2 of the piglets. Then you might miss the virus, although it really caused the infection and the abortion.
Very good. From our conversation with the experts here about high virulence forms of the PRRS virus, it's clear they occur worldwide, known today in North America and Europe as well as in Asia, but still rather the exception than the rule. Nevertheless, some changes seem to be occurring which are being identified by the pathologists. Current research indicates, for example, that these high virulent strains can also induce neurological signs, which was not the case in the past with our experience with PRRS. And we are hearing also that if checking for strains on a farm where suspected occurrence of high virulence strains is the situation, we should be checking for PRRS also earlier than in the past, before the 3rd trimester of gestation. Also, on a practical level, if you're looking for PRRS in fetuses, we should be looking in the thymus. And it's the easier sample to take. And if we can pool 5 samples to run on PCR, that should give us an effective answer. You have been listening to a Meet the Expert podcast presented by Boehringer Ingelheim. Please note that other podcasts in this series are becoming available. Stay tuned and thank you for listening.